ASMR, back with another video. Today we're going to be talking about glycolysis. This time we're going to be using the Stanford metabolic map to analyze the different steps of glycolysis. So, without further ado, let's get started. So this is a Stanford metabolic map, and in order to find the glycolytic pathway, you just have to find this particular area of the map and then zoom in. And you can already see that we have all the different enzymes that are part of glycolysis from top to bottom. And so we can start from the top with glucose, which is obviously our substrate for glycolysis. Okay, and so with glucose, it is the starting point for glycolysis, which is basically a metabolic pathway that breaks down glucose into your pyruvate molecule that we see over here. We also end up producing ATP and NADH, and it occurs in the cytoplasm and does not require oxygen, which is why glycolysis is an anaerobic process. Overall, it is important to understand both the enzymatic steps and the regulation of glycolysis. So let's go over the specific parts of glycolysis. So once again, glycolysis occurs in the cytoplasm. The input is one glucose molecule which contains 6 carbons. You also have to input 2 NAD plus molecules and 2 ADP molecules, along with 2 phosphate groups. And then the output is, the net output is 2 pyruvate molecules, each containing 3 carbons, 2 NADH molecules, 2 ATP molecules, 2 water molecules, and 2 H plus ions. So we can see that right over here. And so when we scroll down to the very first step, the first 5 steps are basically the energy investment phase, and the next, or the subsequent steps, are known as the energy payoff phase. So let's begin with the first step, which is the conversion of glucose into your glucose 6-phosphate. This conversion is facilitated through the enzyme called hexokinase, or if the glycolysis is taking place in the liver or the pancreas, the glucokinase will be used instead. Now, the cofactor for this enzyme is magnesium, and in order to facilitate this conversion, 1 ATP molecule has to be consumed. In terms of regulating this step is inhibited by this molecule called glucose 6-phosphate. This is also known as product inhibition. And then the glucokinase is also regulated by insulin instead of the glucose 6-phosphate. So remember that the glucose 6-phosphate inhibits the hexokinase activity through product inhibition, while the insulin is responsible for inhibiting the glucokinase, or at least regulating glucokinase. And then one clinical correlation to think about with this step is that glucokinase mutation are linked to MODY, or maturity onset diabetes of the young. Okay, let's move on to the next step, which is the conversion of glucose 6-phosphate into your fructose 6-phosphate. This particular step is carried out by the enzyme phosphoglucose isomerase. And unlike the first step, this step is reversible, which is 1,6-bisphosphate. This time, the enzyme involved in this conversion is your phosphofructose kinase 1, also known as PFK1. The cofactor for this enzyme is once again your magnesium, and then again, one ATP molecule has to be consumed. In terms of regulating this step, this enzyme, phosphofructose kinase 1, is activated by your AMP molecule along with your fructose 2,6-bisphosphate that we see over here. So, this particular molecule, fructose 2,6-bisphosphate, will actually upregulate the phosphofructose kinase 1. On the other hand, when you have accumulation of ATP or a citrate molecule that we see over here, so here we have our citrate molecule, the accumulation of that, or I should highlight the citrate molecule here instead since we're still in the cytoplasm. But anyways, the accumulation of citrate molecule or your ATP molecule is going to downregulate or inhibit the phosphofructose kinase 1, which is ultimately going to lead to decreased conversion of fructose 6-phosphate into your fructose 1,6-bisphosphate. This makes sense because when you have enough ATP and citrate molecule, that is a sign that the body has enough supply of energy source, and so there's no need to further break down glucose. Instead, we can convert glucose into its stored form like glycogen, which I'll talk about in a future video. But anyways, what I want you to take away from this is that the regulation of phosphofructose kinase 1 is a critical step in glycolysis. That is why, clinically speaking, it is known as the rate-limiting and committed step of glycolysis. Okay, let's keep moving on to the next step, which is the conversion of fructose 1,6-bisphosphate into your glyceraldehyde 3-phosphate or your dihydroxyacetone. So the enzyme responsible for this conversion is known as your aldolase. And we can see the bidirectional arrow here indicating that this particular step is reversible. So you can either produce your dihydroxyacetone phosphate or your glyceraldehyde 3-phosphate. When you produce one of these products, you can utilize the triosphosphate isomerase to convert into the other product that we see over here. So, and then again, this particular conversion process is reversible. Now, what's significant about this part of the glycolytic pathway is that it signifies the conclusion of the energy investment phase, where we needed to consume ATP to generate our intermediate products. So at this point, we have two G3P molecules that continue through the pathway, or glyceraldehyde 3-phosphate. So there's two of them, not just one. And that is how we ultimately end up with two pyruvate molecules. So let's keep going. The next step is we convert the glyceraldehyde 3-phosphate into your 1,3-bisphosphoglycerate plus your NADH molecule. This will be needed to fuel the citric acid cycle. Or no, it'll be needed to fuel the electron transport chain, which we will talk about later. But anyways, this process will be carried out by the enzyme glyceraldehyde 3-phosphate dehydrogenase, or HEA-PDH. What we end up producing is two NADH molecules because, again, we have two glyceraldehyde 3-phosphate molecules involved. phosphorylglycerate kinase. In this step, we have two ATP molecules produced because again, we technically have two 1,3-bisphosphoglycerate molecules due to the presence of two glyceraldehyde triphosphate. So here it is also reversible, and this is known as substrate level phosphorylation, where we produce the ATP at this step. The next step involves the conversion of your 3-phosphoglycerate into your 2-phosphoglycerate that is carried out by the enzyme phosphoglycerate mutase. Again, it is reversible. Moving on, we have the 2-phosphoglycerate that can now be converted into your phosphoenolpyruvate molecule. This time, the enzyme involved is enolase. What's special about this enzyme and this step is that a water molecule is released. And again, it is reversible. Now we have phosphoenolpyruvate molecule. And once again, remember that we have two of these molecules. So the final step is to convert your phosphoenolpyruvate into your pyruvate molecule. This is carried out by the enzyme pyruvate kinase. And once again, we produce two ATP molecules because we technically have two phosphoenolpyruvate molecules. In terms of regulating this important step, this step is activated by the molecule fructose 1,6-bisphosphate that we talked about earlier, right over here. So the accumulation of this is going to upregulate this pyruvate kinase enzyme, which makes sense because we don't want this to accumulate too much. So we need to make sure that more of these intermediates are being converted to pyruvate so that we can consume more of the fructose 1,6-bisphosphate. On the other hand, the pyruvate kinase will be inhibited by your ATP molecule along with the amino acid